Okay. I just want to start with a few clarifications. I received a message from someone asking, like, if you're doing the Python tutorial at the start, the first sort of practical, which isn't actually a practical, it has these points attached to certain exercises. So none of the things that you will see, like in terms of points, will mean anything in terms of grade, right? So this is all, the practicals are, we're taking this very literally, are for practice. They're not being graded, right? So if you see that, that's just a remnant of when they used to be, yeah? So that's for the Python tutorial. It also holds for the rest. So there are now 3 tutorials already online. You can start working on them. I think I still have to upload the data for one of them, but you'll be able to find that ASAP. So you can work on those on your own, and then from next week we'll have these bi-weekly Wednesday morning practical sessions where you can come here, ask questions. So you can think of those more as like, elaborate question hours, and we'll also be online on Zoom. So we'll make some breakout rooms so you can like collaborate and things like that on there. Yeah. Now, specifically, also, we have these separate practicals, and they are sort of subdivided or divided into different practicals on the basis of topic. And what that means is, we were going to have 4 of these. We're adding a new one, which I think is nice. But they're not really sort of stuck to the specific Wednesday morning practical, right? So you can do these like in reverse order or anything like that. The Wednesday morning sort of practicals are just for us to help you along with any of these. Is that clear? So they're not tied together in any way, these practical notebooks and the practical things in the roster. Yeah, that's clear. Great. And now we are sort of. So I'm talking a bit about how. So I'm teaching the lecture material, of course, and I'm trying to supplement that lecture material with these YouTube videos that I already pointed to. Like I'm thinking many of you have gone to that YouTube playlist and seen what it's more or less about. I don't care too much about all of those things, especially the latter portion, which is about across subject statistics. I feel like that is something that if you end up doing an fMRI experiment, you'll be doing that on your own at some point. I think it's uninteresting, and I think for a brief course like this, I should emphasize other things. So that's what we're doing. So what I've done now is, in terms of the timeline for the course, at the bottom of the syllabus, I've outlined what is expected of you on each week, and I'm sort of cutting it off right now at the end of week 4, because that's when we'll have like the sort of basic theory part done, and we'll start doing things like guest lectures of people presenting their own research and that sort of thing from that point onward. And I'm still scheduling those because they're like friends of mine that are equally busy and so forth and so forth, right? So from, let's say, week 4, so starting last week of February, for example, you'll start working on the project and reading literature that's specific to the projects. Yeah. Any questions about that? Clear? I hope it doesn't feel like too much already. No fearful faces. Okay. Cool. Yes, the guest lectures are part of the exam material, but I won't go into detail, right? Yeah, and the exam. So there's questions on the chat in Zoom. Question is, guest lectures, are they part of the exam material? Answer is yes. And second question is, will the exam consist of programming questions as well? It will not. Yeah. So I'm not, so either you just came from a programming course in the psychology master, in which case I'll trust you, and or you're an AI master, in which case I'm going to bet that you can program, right? So those are the two options. I'm not going to test whether you can write a for loop or whatever. Yeah, and so someone's remarking that it's not very clear which articles have to be read when in Perusall. So this entire schedule hasn't been, set in stone on Perusall, but the first couple of weeks, the first couple of articles are assigned. So you have deadlines for the first couple of papers that you need to read, and I'll be updating that as we go along. Yeah, so you'll be able to read up to 2 weeks in advance. Is that good? Clear? Great. Yeah, that could be. Yeah, I don't forget these things. Yeah. Also, again, about these assignments in Perusall, I'm not like grading whether you read the thing in time, right? This is meant for you as a sort of backbone. Like I'm trying to sync the PDFs, the articles, to things like the YouTube channel videos and to the lecture content, such that, if you follow the assignments deadlines, you'll be in lockstep with the rest of the course. But, if you feel free, if you want, you can randomize this for yourself. And, if you don't care, then I won't care. Yeah. It's all about, what makes you learn best. For many people, that is more structure. So if you want that, then follow the assignment schedule on Perusall. Yeah. Yep. Any more questions? No. Okay, then we'll slowly start with an actual lecture. How about that? And again, I sound like I'm sick. I am a bit sick, but I do not have corona. Whatever that tells you. Okay, I've already told you about what we are working with in fMRI is this really weird thing. It's something that doesn't necessarily relate to what our neurons are doing, but we're looking at some sort of metabolic response, right? And we talked about that on Wednesday. Today, I want to go into how that property was discovered, how we found out that, hey, we can measure with this MR machine something that tells us something about what the brain is doing. And my goal for today is to go through the history in terms of how this was discovered, and then into where we are today. Like, yeah. I am recording this. Yeah. So, So another point, like a practical point, so it takes a while for Zoom to convert these. I get an email and then I have to upload it to YouTube, and I do that. And then when I did that yesterday morning, somehow YouTube crapped up the video. It didn't work for many people. I don't know if you've noticed this. Many people couldn't play the video, so I re-uploaded it. Apparently now it works for the second one that I uploaded. Whenever this happens, I will also upload the video of the lecture to Canvas, right? But we have limited space on Canvas, so I want to, make sure that I don't fill up Canvas. So I'm using YouTube as a proxy for that. Yeah. But the goal is to have these Zoom recordings online either the same day or the next day, but as soon as possible. Yeah, so you don't have to be here. You can leave if you want. Yeah, so this bulge response that we are looking at, this thing that we are measuring. I gave a lecture about neural recording methods yesterday to some bachelor students, and really the point there was with any way that we have learned to measure what the brain is doing, it's not that like nature wanted us necessarily to measure brain activity, right? And so what that means is that for any method that we have that we use to record brain activity, it's based on some weird principle that accidentally allows us to record something that is a proxy of what the brain is doing. This holds for EEG, this holds for MEG, this holds for like anything that we do, right? We aren't necessarily meant to record from the brain, right? And the same goes for MRI. And I hope that you'll get an idea of how sort of happenstance this is, this idea that this fact that we can even record things from the human brain, how happenstance that is. So I want to explain what we're measuring with BOLD, with the MR scanner. So I'll start with a bit of history and a bit of background. So if you think about how fields of science start out, it's usually some weird discovery that people, when they do the discovery, they don't really realize what it means, right? But then someone else comes along and says, hey, but if this is the case, then I should be able to ... right? And then that's the start of something new. Yeah? And you can refer to this as serendipitous discovery, right? So usually you just find something and then you go like, hey, what's this? It also means that people are building on top of one another's serendipitous discovery. So you get really contrived things, or really complex chains of logic, so to say. So, for example, you're able to, if you look at bioradar or something like that, if you were to make a new bioradar, one of the really good ways of measuring precipitation isn't necessarily like the Doppler radar that they're using. You could also use the fact that if you ping the signals between cell phone towers, you can actually measure whether it's raining or not and what type of rain is falling between those cell phone towers. And because the country is covered with cell phone towers, you should be able to measure precipitation and actually create a map of where it's raining in the entire country if you're able to ping the cell phone towers, right? But think of it, these cell phone towers aren't there for the reason of measuring precipitation, right? It's an accidental byproduct of the fact that we all want to have an iPhone in our pockets. Yeah? So this is what happens all over the place. Yeah? And if we want to do something specific, like a specific scientific measurement, we try to be as smart as possible in using this type of thing that already exists. Yeah? And we try to tailor what we do, the measurements that we perform to this situation. So in many cases, this involves just knowing the physics behind a certain phenomenon, right? So that we can use that to measure some phenomenon of interest, right? That's why, because we know the physics, we know that we needed to place the Webb telescope like 1.5 million kilometers away, sort of thing, right? That you tailor how you measure to the physical principle behind a phenomenon. And so that's also what happened with fMRI. Yeah, there is, so people discovered in 1991, there's actually a few people that discovered this more or less around the same time. Discovered that you can do functional mapping of the human cortex while the people are alive, well, in an MRI machine. Yeah. So this happened at a point in time when people were already doing MRI for anatomical things and clinical situations, right? But this is the first discovery of people actually being able to measure function in the brain. Yep. And so we started out being able to do fMRI with contrast agents. So what you do with contrast agents, you inject something, it goes into the bloodstream. And then, so this happens in angiography, for example, if you really want to image the vascular structure of something, then you do that. And then you can image exactly where are the blood vessels. But you can also just inject other things that get taken up into the tissue, for example, and then image those, like a staining technique sort of thing. And people started out in MRI using contrast agents. And in that case, it's really similar to PET. Who of you have heard of PET imaging? Yeah. Okay, so I won't go into detail about PET because like it's old school. And it's not, yeah. So people used to be, so there used to not be fMRI, right? And around that time, like in the 90s, early 90s, people could do functional imaging using PET. And PET is positron emission tomography, which means you inject a radioactive tracer into someone. And if you label if you make that radioactive tracer, for example, glucose, then all of those radioactive molecules will start piling up in regions in the body that use a lot of energy, right? Use that glucose, and you can image that. You can image where does the radioactivity come from. Yeah, you had a question?
Yeah, how would you use that in the brain? Because brain consumes a lot of sugar, if not more than the heart. So
yeah.
Does it apply in the brain?
Yeah, so for brain imaging, there were two ways of doing PET. This is your question about PET, right?
Yeah.
Yeah. So there's two. You can label glucose and you can label oxygen. So you can do either. And in the case of oxygen, of course, you have to have people inhale this radioactive gas. And then it ends up where, well, more or less the same as glucose, right? It's the oxygen that burns together with or that burns the glucose. Yes, and indeed, you do see that glucose or oxygen pile up in different places in the brain. So if you or in the body. So if you do a whole body PET scan, there's actually the best PET scan center of the country is here at the VUMC. I actually worked there for a while, and you'd be able to see these whole body PET scans, and they are really exquisite and quite beautiful. What you see is generally the brain and the spinal cord. You just see that lying inside someone's body. So you see more or less the whole body because it sort of shimmers a bit, right? There's a very low baseline. Every cell in your body uses glucose, so it ends up more or less everywhere. You see the brain because it uses 20-25% of your entire budget, right? And then you see the kidneys because that's where all of the byproducts end up. And then in many cases, you see, well, the muscles a bit, like you said, the heart a bit, but not that much. And generally, this is used in oncology because like cancerous tissue doesn't have a metabolic break associated with it. So it actually pops up really, really strongly in these types of scans too. So you will be able to see metastasization, for example, very easily. So it ends up everywhere. Yeah. Yeah, so if you look inside the brain, of course, then you can have someone perform a task where they are looking. So there's a famous experiment where it's a PET study, and people were shown still images, right, in 1 minute, and then another minute they were shown moving images, right, and then they would do this like 1 minute of still, 1 minute of moving, 1 minute of still, 1 minute of moving, and so they could look at the image taken during that still minute and the image during the movie watching sort of minute, and they could subtract the two, right? And that allows you to see what brain activation is different between the two, right? And so this allowed them to map out a motion selective region in the human brain. So this happened around 93, 94. So people got famous because, hey, this was really like this in the 90s. What time was that? Like Bill Clinton and stuff, right? So around this time, people were starting to be able to like look inside the functioning human brain, and this finding of this motion region in the human brain was one of the sort of pinnacle findings. Yeah, but I'll get into the logic of this type of analysis and this type of experiment much more deeply later.
In the fMRI, we don't without the contrast agent.
Yes.
So why do we use the contrast agent?
Yeah, good question. Like, why do you, right? Yeah, usually you don't, right? But what happens is, I'll just, let me do my story and everything will be clear, because I actually have a narrative arc that actually treats this topic. Yeah. So in the clinic, people do use contrast agents quite a lot. Oh, yeah, sorry, I will repeat the questions. I now forget what they were, but I'll do that from now on, okay? So gadolinium is a contrast agent that's used a lot in the clinic. You can inject it. It's a metal. It is actually, so because of its specific chemical and physical properties, it is the most paramagnetic element at body temperature. Right? It's the most, yeah?
What does para in the paramagnetic mean?
Yeah, I always forget this. I'm not like an MR physicist, but yeah.
I think I googled when I was is either para or diamagnetic.
Yeah.
About how the things are going in the other, if it's going in or comes opposite way of the magnetic field.
So the question is, what's paramagnetic? And paramagnetic is like the opposite of diamagnetic. And I always forget, one of the two really lets through all magnetism and the other one blocks all magnetism. And I think paramagnetic blocks the flow of the magnetic field lines, and diamagnetic lets them through. Am I right? Okay. Yeah. So this is not going to be an exam question. Just having reconstructed this, yes, of course, paramagnetic means it obstructs, so it distorts visual fields, magnetic fields. Yeah. And that also means that you can, that property means that we can start to pick it up if we have a huge magnet that we use to image the body. Yeah. So if you inject gadolinium, it will go through the bloodstream and give you a way of imaging how much blood volume is present in a given voxel. So we already talked about the voxels, right? That's clear. And so what in essence you're doing is you're making measurements of the cerebral blood volume. And if you do this over time, of course, you get a time course of cerebral blood volume. So I'm just going to highlight what they did in this first fMRI experiment that used this contrast agent. What they did is they had this local radio coil on the occipital lobe, it's the back of the head, and they were then able to construct these images. And they were then able to make these measurements of blood volume. Then what you can do is inject, and then you see this bolus of the gadolinium go through the bloodstream. Yeah. And you can do that when people are resting. In this case, that would be like looking at a blank screen or having their eyes closed, right? Because we're looking at visual cortex, so we want to either activate or rest visual cortex. And the opposite thing that they did was activate it. I don't know exactly the visual stimulus that they used, but we know that this part of the visual stimulus really likes it when you flash stuff of very, very high contrast and flash it really fast. So people usually take a chessboard and then rotate it 90 degrees, show it again, rotate it 90 degrees, show it again. So you get like black, white sort of flickering going on. And if you do that and people like actually made videos of this, if you do that at exactly 8 Hertz, it gives you a very, very strong bulb response in the human brain. So if you're interested in this part of the brain, what you use, and it's really boring, but all of the researchers use this checkerboard that counter phases, as we call it, at exactly 8 Hertz. And that gives you an enormous blood response in the human brain. And so my guess is they're more or less using something similar to that here. I haven't looked it up. And what you're going to get is this response to the injection that then differs for activation than it does for rest, right? So the shape of this bolus is there because you have this wave of gadolinium going through the bloodstream. Right? That's the reason why this is already deflecting. And so because it's I'm slowly reconstructing this. I'm a bit slow today, so my apologies. But because this is paramagnetic, what you see is that this is going down, right? So it's decreasing the signal, the magnetization at that point in the brain. Yeah.
Is with resting mean that you do not give stimulus?
Yeah. So there's two stimuli here, if you will. There's a gadolinium stimulus, right? And that causes this boom, this downward deflection. And then there is a visual stimulus, right? So the visual stimulus is just the subject looking at the screen, and the screen is going bam, 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 like, or not, right? And so you're always going to have this effect of the injection, right? And then the difference between, like I just said also about the motion and stationary thing, right? The difference is due to rest and activation gives you the actual change in blood volume that occurs as a result of that visual stimulation. Yeah, and this is akin to the Donder's subtraction method. It's really old. It's like the simplest experimental paradigm that you can do. You just do exactly the same thing, but in one case you change one thing. And you can do this to map out motion areas in the brain. You can do it to test whether you can actually measure activations in terms of blood volume. It was invented for reaction time experiments. It's in essence the simplest experiment that you can do. It's boring. Yeah. But here you see that you actually have a lot of you can cause a lot of signal changes in an fMRI experiment. When you inject gadolinium, it's a very strong effect, right? And you actually see that there's quite a strong effect of this change in blood volume. See that? That's not tiny, right? What you also see already is that something happens in terms of this timing. So first you get a dip, right? So this is smaller, and then here it becomes larger. And this shape of that difference time curve is something that's interesting, and we'll talk a bit about that later on. Yeah.
Why do you if they have their eye closed, or why is their brain signal intensity?
Oh, yeah, so that's a good point. It's very clear. Let's keep this very clear. That's because of the injection. Yeah. So the injection is already causing something. Yeah. And so it's very nice that you get a very strong signal because of the injection, right? Because you have something to hold on to. At the same time, you notice it sort of complicates the logic here, right? Having this injection play a role there. So yeah, you can see how this was, think about that time. I don't know if any of you were born then, but Bill Clinton was president of the United States. People were really like, stuff didn't exist, like cellular phones didn't exist, right? And then this was the thing that happened that really shook a lot of scientists, thinking like, hey, we can start measuring brain activity in human beings. And so this became a thing. That also, whenever something like this happens, a lot of people start jumping on the bandwagon, right? Once people realize that this sort of thing has become possible, that's when they start going for it. And so they started to think like, okay, how can we improve this? Because there are definite downsides to using a contrast agent like this. It's a metal and you're injecting it into a person. Yeah, well, you get the obvious downsides, right? Anything invasive is highly limited, right? If you want to do it like once for oncological things, right? So similarly to the PET thing that I outlined, you can use this in oncology or angiography. You can make things very sensitive, and that's cool for a one-time measurement, right? So in the clinic, this is awesome. Yeah.
What is the first one of the two? And
angiography is the imaging of the vascular structure of the body. Yeah. Yeah, so you have, if you use, there's other contrast agents that you can use specifically that do not permeate well, I think gadolinium also, but you can make these really beautiful, like images of the vasculature of someone's body. Did I show you that that vasculature of the brain at some point? Yeah, I forget. Yeah. Actually, so gadolinium actually it goes into the blood and then it goes out. That's why you get this transient curve that I showed you on the previous slide, right? There are other agents that stay in the blood for much longer, and those are used, for example, in animal fMRI a lot. So there's this manganese iron oxide particle thing that you can inject. It stays in the blood for quite long, and it actually increases your signals very, very strongly. So people use that a lot. But as you can guess, in the case of both this gadolinium and this myon, you can't just keep injecting people, right? That is just not what you do, because you know, that's what the thing that you would want to do for these cognitive neuroscience experiments. They don't serve a clinical purpose. There's no, you know, real upside for the individual to these types of things. You can't keep doing that. Yeah.
What's the difference between manganese and iron and gadolinium? Say that gadolinium goes away from the blood and they stay in the blood.
So yeah, I don't know exactly the biological principles like for a fact, but you can imagine that gadolinium is just really like a metal particle, right? It's tiny. And this myon are actually pretty big particles. They're actually like little balls. And so I think this is not cleared as well by the kidneys and the liver. Yeah. But nonetheless, you don't want to keep exposing people to this type of thing, right? And so there's fire apparently. Yeah. Oh yeah. This is lasting way too long. So you can imagine that people were stuck with these contrast agents, and then this paper came out showing, hey, wait, this is based on blood oxygenation, right? So we can do magnetic resonance imaging based on the blood oxygenation. Wait, I don't have to inject people with blood, they already have it, right? So it turned out with this article that we actually have a contrast agent of our own in our bodies, and it's based on blood oxygenation. Yeah, so you see that all of these negatives of having to use these invasive injections just fall away immediately, right? And you can see how this duo of discoveries, first, hey, we can image the human brain. There's still a little downside, like people, you know, you have to like keep injecting them. At some point, they're likely to get sick. And then all of a sudden, someone comes along and says, hey, but you can do this based on the blood itself, right? And then that opens up all the doors. Okay? I just want to, like, I think it's always important to try to look back and imagine how people were feeling about what happened during any point in time, especially when it comes to the evolution of science, because it tells you a lot about how this works. So the solution to this invisivity problem is that we actually have an endogenous contrast agent. It turns out that if you look at the magnetic properties of blood, these are heavily influenced by its oxygenation. And it turns out that if the hemoglobin in the blood carries a lot of oxygen, it is diamagnetic. And when it is deoxygenated, it becomes paramagnetic. Yeah. So it becomes a bit like gadolinium as the oxygen is extracted from the blood. You know, when that happens, it's when things are active. Right? So that's pretty awesome, the fact that this is the case. But of course, then we still have to measure it. Can we measure it? Well, it also turns out, happily, right? There's cell phone towers, so we can measure rain, apparently, right? Turns out that there is a specific MR contrast, a specific sequence that we can play on the scanner that reads out the tissue properties in exactly some way that is sensitive to this difference in oxygenation of the blood. I'm just showing you like a T2 star weighted anatomical of my own brain, because it's, you know, it's also used as an anatomical sequence. It's just that you can also try to acquire it over time, and then you actually get functional MRI. So we're in this lucky situation where we have a, yeah?
Yeah, you said anatomical sequence.
Yeah, so you have functional MRI and anatomical MRI, right? So anatomical MRI really is, a single 3D image. Okay? And a functional MRI, because function expires over time, right? It unfolds over time. You need to do multiple measurements over time. And of course, then you need to have an image that is actually sensitive to something that correlates with function. And here we have it, right? So this specific MR contrast. We'll have a physics lecture at some point, and this concept of contrast will become much clearer. So this is a specific type of using the scanner and reading out the signals that pop out of the brain that make you sensitive to specific types of tissue properties. And specifically, that tissue property is related to this oxygenation of the blood. Right? So this gives us this, you see how this is a very convoluted proxy of what you might be interested in, right? And this is important, I think, that I really want to drive that point home over and over, right? It's an accident that we can do this. It's a complete accident, right? If we didn't have the scanners already in the hospitals, if we weren't able to really understand the physical principles of magnetism in tissue, if this didn't accidentally turn out to be the case, and if neurons didn't, in a faithful way, also cause changes to the blood response, then we would be nowhere, right? So it's all an accident. That might not sound very hopeful, but it is very lucky, right? It's a lucky thing. Now, we already talked about the paramagnetic diamagnetic, right? Paramagnetic is it sort of obstructs the magnetism. Very qualitative way. Thanks for, yeah. It turns out that when you extract oxygen from the blood, it becomes more paramagnetic. It becomes more like gadolinium, right? And you knew from that injection of the gadolinium that the signal went down, right? And so what you expect is that these values of these T2 star values, they actually decrease as a result of neural activity, right? Because neural activity will draw oxygen from the blood, right? And thereby deoxygenate it, right? So what you expect, if I put you in the scanner, measure this T2 star value, right? Over time, you activate, it should go down, like we saw for the gadolinium, right? Right.
So do you see anything where there's no activation?
There is never no activation unless people are dead.
Oh, so it's, okay, so it's always, but then some are like more activated than others, and therefore those are more.
Yeah, so we'll get into this in a later lecture, but your brain is always doing a lot.
Okay,
Yeah.
Makes sense.
So there's no, yeah. Well, you could, again, this would be invasive. You could like anesthetize people and then wake them up and anesthetize them and wake them up, and that would be large changes in the brain. But you're not allowed to do that. So yeah. Okay, so we're clear, right? We expect neural activations to cause decreases in T2 star values, right? So if we measure those, we should see these decreases and we're good, right? Yes. You use the scanner slightly differently, but they are both based on the T2 contrast, which in the physics lecture we will explain. And so T2 star isn't necessarily, so it's very sensitive to, well, the physics lecture will explain in detail, yeah. And our physics is kind of complicated, so I would likely get into a swamp if I started talking about it now. Okay, so we're measuring this blood oxygenation level dependent signal. It's actually 45 already. Should we have a break? Let's have a break. 5 minutes, 10 minutes, something in between. Yeah, I'll start talking again at some point between 5 and 10 minutes.
Over dit onderwerp heb ik daar van ik ben reuzenzaak. En hoe kunnen we bijvoorbeeld het eisen weer afstuderen?
Ja, ik begeleid elk jaar wel 2 of 3 AI-studenten in een onderzoek. Dus dus thesis en internships, ja.
Oké.
Laat me weten.
Okay, I'll just start talking again. I just noticed I'm only at 1/3 of my intended lecture. So we're gonna have to get going. Fire it up, that sort of thing. Yeah. So I hope that you start to get an impression for how impossible it was in, let's say, the 1980s to look inside people's heads, right, and see what their brains were doing. And it turns out that around this time, people started to have these discoveries that started to sound like, hey, we might be able to start doing this stuff with, looking inside of someone's head. And at first there was this invisivity that was problematic. But then this discovery that I showed you on the last slide showed that, hey, even the blood can function as a contrast agent. We have, most of us, plenty of blood to go around. So, you have a lot of contrast agent to work with. And then the next step was this one, where they really showed that if you do primary sensory stimulation, again, vision, you actually get, this is the early one, sorry, I'm gonna get, yeah. So now we notice that we can do this non-invasively too. And we just talked about how, so here you see this off, on thing for people being shown a visual stimulus. Here you have the back of the head where the visual cortex is. And you see this activation versus nothing, right? Is that gorgeous? It's similar to that thing that I was talking about in PET. You can actually do it in MRI too. Magic. People didn't realize that you could do this at all. And now all of a sudden we can look and see the human brain, a live human brain function. Yeah? But there's something weird here in this image. We see on, bright spots, right? And then off, nothing. And I don't know if you still remember, because we had a break in between, of course, things fade incredibly quickly. But we just discussed that the signal intensity should be going down. And it did what? Like what? That can't be the case. This is wrong. And if you look at what's happening here with this photic stimulation, just means you show people stuff. And if you look at the signal intensity there in those regions, it actually goes on, off, it goes up during on. So we're wrong, right? And there we are. That is strange, right? Because we just thought we have an internal contrast agent. It is paramagnetic. As oxygen gets depleted because of neural activity, the signal should go down. And here we are at a complicating factor, right? Yet another one, right? We found this beautiful thing. We can use it. Everybody wants to get on this bandwagon, and interpretationally, we have a slight problem. So let's dive into this a bit. Why do we get this positive bulge response? Well, it turns out that when neurons activate, they cause changes in not just the oxygen content. Maybe at the moment that you die and then you have neural activity, it would just decrease the oxygen, right? But of course, you have a body that's alive and that doesn't stand for just a decrease in oxygen. It will, it wants to keep its metabolism going. It needs to supply a new oxygen stream. Yeah. So there is this balance, right? So what does a change in neural activation cause? Well, it causes many different things, right? It causes This changes the metabolic rate of glucose consumption. It's not simple. It also changes the rate of oxygen consumption. Right? And this is just do we have both the consumption and the resupply, of course, in these types of, especially for glucose, right? You can have a local repository of glucose, and then it gets resupplied. So these two things we can measure with PET with these two contrast agents that I was talking about before, glucose and oxygen. You can also measure cerebral blood flow specifically. So that's the flow, the inflow of blood, which is also changed by neural activations. You can look at the cerebral blood volume, right? And that's what we measured with these gadolinium and myon contrast agents. And we changed the hemoglobin oxygenation, right? And that final thing is what we were talking about just now, right? That final thing was, hey, it's going to go down. All these other things, we have no idea what it does, right? And yes, it's really the increase in blood supply. Yeah.
So these are all activities that happen when a neuron is active.
Yeah. So you have a neuron that fires, and then you get all of this stuff going on, right? And this is, of course, something that happens both in the neuron. It happens in the glial cells next to the neuron. It happens in the smooth muscular tissue surrounding the vasculature. It happens in the lining of the vasculature, which changes permeability. The sizes of the vessels change, and the flow of the vessels change. And all of that is happening in tissue that is maybe still experiencing neural activity, and where the blood is being pumped through by the heart, and you have this pulsation going on, right? It's a mess, right? It is a true mess. And so one thing, let me see, where am I? Yeah, so the point is, the measurement that we are taking is very, very complex, and I still haven't told you about this positive response, right? Why does it happen? I'm already showing you the why that is here. So why is that? Well, you have deoxyhemoglobin maybe changing, right? So that's the decrease in this, but you're actually having an overreaction of the vasculature. So this entire sort of cascade of processes overreacts and then supplies an overabundance of new oxygenated blood to this tissue, right? But you can imagine that this is not just a change in the ratio of the oxygenation in all of the hemoglobin there. You know, this comes from somewhere. It is supplied by blood flow, right? And it's only possible by changes in blood volume. Right? So there is a lot of stuff going on, and then the total sum of this is this positive bold response that we measure. Yeah, is that clear? So that actually just switches the sign.
Does the total hemoglobin also increases because of the overreaction?
Yes, yes. So what happens is you might have not so much blood in a certain location, right? You get neurons sorry, activating there, and that will slightly decrease the oxygenation, right? For just 100 milliseconds or so. Then the tissue responds, the vessels open, and new oxygenated blood comes in. And so you can imagine that that is just an enormous wave of extra oxygen that then changes this ratio between oxygenated and deoxygenated blood. And this is very complex. It is an active area of research in and of itself. But it just goes to show, like with the cell phone towers in the rain, we are measuring something that, we aren't intended to measure necessarily, right? We have to understand that this is the case and then take that into account. Now, that's, yeah.
Sorry.
No.
You can go with one more slide. Like, I don't understand the last sentence, because like more of the oxygenated blood comes in, but that's not the peak, right? Or is it?
Yeah, so so what you get is, I'll go into the shape of the bold response and how we use it in analyses later. So I'll get to that. But for now, just take away from this that what you're measuring is a positive response instead of a negative response because of this oversupply.
Okay.
Yeah. So where you would expect a negative response, now we actually measure a positive response, and that's because the tissue actually inverts what we thought we would be seeing. Yeah.
Coming back in more detail.
Yes.
Okay.
Yes. But it is really, it's really interesting. But I'll talk about the shape of the bulge response more, and then this will actually come in. Like we're going to look at this time course in more detail in a later lecture. Then there's the question, okay, but we have electrical activations that we're interested in, right? We'll generally think of electrical activations as the true neural activity, right? And not the blood thing that we're measuring. But how does the blood flow, the bulge response, how does it serve as a proxy of electrophysiological responses, right? So, or any other measure for that. So how do we interpret this, right? What does it mean to measure a bulge response if what we're interested in is actually the electrical activations of the brain? And so let's go into that a bit. And so let's talk a bit about this relation between bulge and, in this case, specifically electrophysiology. So let me highlight a few things that we can get from electrophysiology. So we can record with an electrode or with a magnetic apparatus, we can record the electrical activation of these cells. These are pyramidal cells. They're shaped like a pyramid, hence pyramidal cells. And they are usually aligned like this. And if you look at the cerebral cortex, so usually when we talk about brain, there's a lot of brain that's not the cerebral cortex, but usually when people say brain and they start talking in detail, they'll be talking about the cerebral cortex, the outermost part of the brain. And that's this convoluted structure where you see cortex and white matter. I already showed you this in the T1 and T2 images, right? You can actually see the difference. This is the gray matter surrounding the white matter. And so all of these pyramidal cells are oriented like this, like this, like this. And when they fire, they usually fire in conjunction. And so what they will do is they will create an electrical field in this direction or in this direction or in this direction. And you are able to pick it up with electrodes or magnetic field devices, either on the scalp or directly onto the brain if you're doing intracranial EEG, or if you're doing invasive electrophysiology, you have an electrode, and that will capture the electrical activations. And you can actually have electrodes with multiple contact points, and there's beautiful evolution of technology in terms of electrophysiology. It's insane what people can do. They're starting to create electrodes that are created using all these new semiconductor fabrication techniques. So they can actually create electrodes with thousands and thousands of contact points. So if you lower this into the brain, you actually have incredibly high resolution sort of electrical images of what happens in a local piece of the tissue. That is an awesome, that's now happening, and people that's really a new wave in electrophysiology. But I'm digressing. One of the things that we measure when we measure electrical fields, like voltage changes at a specific electrode, regardless of where it is multi-unit activity. That is, the tip of the electrode is, oh wait, I got questions. Oh yeah, deoxy. Oh no, this is, I think, the signal that's generated by the deoxyhemoglobin. So this is still about the last slide. Yeah, and I'll try to remember that. I'm sorry. So the tip of the electrode is going to be close to a bunch of cells, right? And it's really hard from the signals themselves to find out which cell is firing which. So what that means is we are always looking at some, multiple of units. Yeah. And if you look at just the very high frequencies in the electrical signals that you're recording, what you're picking up is the spiking of a bunch of cells. So that is the activity of multiple units standing for, that's what MUA stands for. Yeah?
You said that it's very close to the cells, so the electrode is actually going into the scalp as well.
Yeah, so you do this MUA thing with anything from ECOG to invasive animal stuff. Yeah. So you only have these types of data in monkeys or cats or mice or whatever, or in epileptic patients that are receiving surgery to find the locus of their epilepsy. And then people would use this ECOG thing. Yeah. So in the case of, you would not wish this on anyone. Like in the case of this epilepsy, if you have intractable epilepsy, what these surgeons do is they actually cut away a window of a pretty large window of your skull. They lift it out. They put in a net of electrodes on really the brain tissue. So not even the brain, the dura, like the fleeces around it, but really on the neurons themselves, like really here. And then start recording electrical activations. Yeah. Yeah, so globally they can tell like, oh, it's in this lobe of the brain, but they need to find a very specific portion of tissue that they can resect to make sure that the epilepsy doesn't reoccur. Yeah. That's a separate lecture though. I can talk about that at some point. We're doing this type of research ourselves also, so yeah. But so any electrode doesn't only pick up very fast signals, you're also measuring slower signals. And these are local field potentials. And the local field potentials are thought to reflect more the input into an area than its output, right? So if we're measuring fast signals, we're looking at the outputs of a specific region. And if we're looking at local field potentials, we're looking at the inputs to that region. And there are different bands of local field potentials that you can also pick up using EEG. So you have slow frequencies, like low frequency oscillations, like signals that you pick up in the theta or alpha band, which is like slow between 4 and 12 hertz. So these names, I won't quiz you on it. It's not an EEG course. And then there's faster oscillations, like signals that you can pick up, and they're called beta and gamma range. And you have to think of like the slower the signal that you pick up, also the larger the spatial extent that it stems from. Yeah? So gamma is thought to be relatively local and reflect more or less something that's as spatially specific as the multi-unit activity, whereas theta and alpha are actually really, really broad, like centimeter scale in terms of space. And then, of course, your question becomes, well, this bold response that we're recording, what does it correlate with, right? Is it similar to multi-unit activity? Is it similar to local field potential? What are we looking at when we're looking at this bold response, right? We need to know this, otherwise we can't actually say anything about what we're learning about the brain. Clear? Yeah. It stands for electrocorticography. Yeah, or people also call it intracranial EEG. Now, some people, of course, if you're really interested in this question, you can't do it in humans, right? You cannot put an epilepsy patient with electrodes in their brains in an MRI scanner. And so people devised a very complex experimental setup in which they could do this with monkeys. And what they were able to do is perform an electrophysiology experiment simultaneously with a bold experiment. So electrophysiology in the scanner. And you have to imagine that you're putting all of this electrical measurement stuff inside a huge magnetic field that is also switching really, really fast, causing all sorts of electrical currents in the electrical measurement stuff and short circuiting. Like this really took a long time for them to be able to do this. By doing it. And what they could do then is show which of those frequencies, which of those electrophysiological signals correlated with the bold. And here you see this is actually more or less the data that they acquired. Here we go. So they're showing again, a checkerboard. Who thought, right? They're doing this in the visual cortex. They're showing a flickering checkerboard stimulus on, off, for 10 seconds or something, and then off, on, off, on, off. Yeah. So the red line is the bold response. And you see that it happens like so. It's a bit delayed, and that's because this blood response takes a while. And then you see that that the electrical signals are subdivided into different bands. We start with slow bands, and then we go to higher bands. And then what you see is that, more or less all of these activations. So here you see, for example, the multi-unit activation going up and then going down, up, down, right? So you can look at the correspondence between these signals, between each of the electrical signals and the Bold response. And if you do that, you find that it's especially this gamma frequency that seems to be the most strongly correlated with the Bold response. Okay? So what that means is a few things. So we also see that it actually is the case that the multi-unit activity also shows something specific about the Bold response. So there are relations with all of these different bands, right? So it's a broad thing, but this gamma range seems to be the strongest. And then we're using the statistics to correct for 1 over f. I won't go into that. We can talk about that later when we're talking about Bold responses. But the takeaway from this is that we learn that the Bold response reflects local processing. Yeah, that is to say, if we find that the Bold response, like we do, we find that the Bold response is most similar to the gamma band fluctuations, these fast fluctuations. It means that we're looking at very local processing, and it reflects more the input into a region than its output, right? If we had seen that it's the multi-unit activity that correlates very strongly to the Bold, then we would thereby conclude that it's the output of a region that is what drives the Bold response. Okay? Here we have to conclude that it's some weird mixture of all of these different input, output, and different scales. But primarily, we're looking at local processing. And that's a good thing, right? Because it means that the specificity that we get from recording fMRI, which can be small voxels, right? That it's actually sampling from the local electrical fields. Yeah? So it's not like there's an inherent blur going from the electrical activation to the Bold response. It's actually very, very local. Yeah? So that's a good thing. We're happy. Yeah, so that's not important. So that's great, right? I don't know about you, but I'm more happy than I was before. Before, I didn't know what specific electrophysiological things related to my Bold response, and now I do. I know that it's local, and that helps me because it tells me that my Bold responses are specific. Yeah, and this specificity is, of course, what you want. The less specific something is, the less informative it is. So now we're looking at this Bold signal and we're thinking, hey, this could be really informative about what the brain is doing. But let's go back to this relation to electrophysiology, because as you could have guessed from the way that I'm talking about this isn't really the final story. And it turns out that recent findings really show that you can get Bold responses actually without any neural firing, without any electrical activation. And the way that people have found this is by optical imaging. So if you take an animal, you can lift the skull, and you can actually just take a camera and point it at the brain. Now, in general, what you would just see is gray, pinkish stuff. But it turns out that if you illuminate that brain with two different lamps of different wavelengths, and you pick up the signal also at these two different wavelengths, and this is again one of those stories like, who knew, right? But you can actually measure the oxygenation of the local tissue by the reflectance in these two different wavelengths, right? So you can actually look at a pixel in your image, divide the red light by the green light intensity, and then get a proxy of the oxygenation at that pixel. So it's, and then again, one of these super weird, happenstance things, but you can measure the oxygenation of the local tissue there. Now, I'm going to quickly go through this incredibly complex graph, but it is a beautiful graph. So what they were able to do here is, of course, they've lifted the skull so they can put an electrode in tissue. I'll stop talking about gory details at some point, I promise. So they can put an electrode and actually measure the local oxygenation at the same time, right? Again, beautiful, but now not as complex as doing it in the scanner. And what they can do is show a stimulus, a visual stimulus in this case. So this is in monkeys, and they're just letting the monkey fixate the center of the screen. So it's looking at a point, and then once in a while there's a stimulus. And this once in a while happens every 30 seconds or so. And what you get is, of course, if you measure the electrical activation and you increase the strength of the stimulus, that's the contrast of the stimulus, what you see is that you get a graded response in the neural firing. If you make a stimulus stronger, you'll get a stronger neural response. Okay? That's clear, I hope, right? No, there's no debate about what LFP reflects. It's the input to the dendritic tree of the pyramidal cells. Yeah. There was a question about what LFP was. So here you see this graded response, right? And you can imagine this like the contrast is the difference between the white and black in a stimulus, right? So if you make the difference small, you just get a gray stimulus. If you make it large, you get black and white, very strong, like those checkerboards, right? It's a strong stimulus, very high contrast. So the larger the contrast, the larger the response. And so what you can then do is say, if I now know the bulge response to a stimulus presentation, then I can simulate what should come out, right? I can simulate what the bulge response reflecting these electrical activations should be. And that's what we're seeing here, right? So, of course, there is no firing for no stimulus. Yeah. And then you have these different contrasts. So you see that this is the bulge responses that we predict, right? And in this mode of measurement, again, a bulge response is negative. That's just the way that it works. But again, we're seeing this whole supply of oxygen type thing. Right, so this is what we expect to see in the signals. Yeah, if we're looking at the bulge responses in this tissue. And then what you can do is then actually perform the experiment. And so crucially, what you expect is this graded response going from gray to black. Right. Here is the measurements. You expect a graded response going from gray to black. Hey, we get that. So there is a reflection of neural processing there, right? We see a contrast-dependent relationship. We find it in the neurons, and now we find it in this bulge response. Great, right? Now look at the blue line. This is kind of problematic, right? We have no neural firing, and still we have quite a strong bulge response, about half of the maximum. Yeah. So the question is, what the is that doing there, right? And these researchers have since started looking at why this is, or where it comes from, and the thought is that this is something that reflects the expectations of the monkey. So the monkey knows that every 30 seconds there's going to be a stimulus. Yeah? So there are these expectations of the monkey, like, now it's going to appear. Right? And then sometimes it doesn't, but that expectation is still there. So the idea is that your brain isn't just doing stuff, or at least the bulge response is reflecting not just your brain doing like local computations that we thought we were measuring with the LFP related story that I just told you, but there's all sorts of neuromodulatory metabolic things that happen when you expect something to happen. You know, this anticipation, when you know that something is going to happen, something's going to happen at some moment, you really expect that it's going to happen, right? There's no stimulus to compute on at that point, but your whole brain is going like, right? Your blood pressure rises to that point. And so that's what we're seeing there. Yes?
Is it also when the negative response already started before the stimuli?
Yeah, that's likely the reason. Yeah. And so they started doing like experiments where they randomize the timing, and they see that this decreases much more strongly than these responses and that sort of thing. But it's it's another caveat, right? Always think deeply about what you think you are looking at in terms of signals. Yeah? You cannot take things for granted in any area of science, really. But I think in brain imaging, we are confronted with this very, very directly. Any questions about this? Yeah. The red line. Where? This one. This is my arrow. I drew it. This is when the stimulus was on the screen, I think. Yeah. Yeah. Great. Now, I want to quickly go into and do so quickly. Oh man, I'm so slow today. I'm sorry. Okay, so the YouTube channel, the playlist that I'm using as a supplement to what I'm saying here is from 2015, and they are treating like they're doing very basic stuff. Right? So if you go through those modules, what they're talking about is really like the state of the art like it was 10 years ago or something like that. And a lot has happened, so I just want to really highlight the things that have improved since that time, because they are there is a very, very quick evolution in terms of what we can do with fMRI. Yeah. So this scanner is very versatile. We talked about that in the last lecture also. And it turns out that by increasing the field strength, and you cannot do that, in the program, you need a new scanner, but they are getting cheaper, and the higher field strengths are getting more and more normal to have. And by improving these programs that you can run on them, there's two ways in which we can get closer to the biological level that we're interested in. And the first is to scan faster. And that's important because I've already talked about, when your heartbeat sends this pulse of blood through your brain, we're looking at a blood response, right? So you're going to get all sorts of weird artifacts from this pulsation. And if you have more information in the time domain, you can actually denoise your signals from this type of effect. And we'll get into how you would do this and all of that, and they're practical about how to do this. So we'll talk about that one. And later we'll also talk about what I mean by the word connectivity. For now, that's not really important. And we can scan smaller voxels. And it's a bit of a hype, but there is a large development there. If we have a good scanner, we can scan voxels smaller than 1 millimeter. The idea is that we can do laminar Fmri in this case. So what that means is, again, here you have your brain's cortex. Again, this is my brain. And if you look at, for example, V1, it's nice and, its orientation is okay here. You can actually take this little column of V1 and look at it. So this is about 2 millimeters. And we know that the cortex is layered. It consists of 6 layers, and it's thought that all of these layers have different computational roles. That is to say, layer 4 is actually receiving the bottom-up input from the thalamus. I'm speaking specifically about V1 here, the first visual region of the brain. But the input comes in layer 4, and then there's lots of computations that take place that go in these directions. Then you get output going from layer 2 to the next visual region, for example, in the case of V1. And these other layers are actually the recipients of different types of top-down feedback, right? The expectations I was talking about just now, all right? When you expect something to happen, that is the result of feedback. Okay? So these different depths in the cortex have different computational roles. And with smaller voxels, we can start to have a voxel here, and a voxel here, right? And we can have independent measurements of these different layers that we know or think have different computational roles. So we can actually use this type of measurement to dive into a new level of the computations that the brain is performing, right? Not at the level of whole brain regions, but at the levels of these layers. And you can see how that is a promise. At the same time, you can imagine that having voxels this small, things get really finicky. For example, you have to make sure that your functional data overlay with your anatomies. That happens sometimes. I have to reprogram that button. So this overlaying of the functional images with the anatomical images, it has to be just right, because if you're wrong by 1.5 millimeters, you've just shifted the white matter boundary to halfway through the cortex, right? And this is incredibly important, and this field is going wrong in many ways. I've also published in this field, and when you look at other people's stuff, you just see that they're doing it wrong. So yeah, that's a bit annoying. So details matter. Is all I'm saying. But there's there's a beautiful promise of this type of imaging, right? Because, like I said, it brings us closer to the biological level. And I don't know about you, I was trained as a biologist and as a physicist way back. But this idea that, you know, your brain is a biological machine, and in principle, if you're coming from AI also, it's a biological computer, right? So we want to try to understand the computations that the brain is performing. And this laminar fMRI actually is a very beautiful way of trying to understand those computations, those biological computations. So I want to highlight, you know, how we can dive more deeply into that biological detail, and that fMRI can actually start doing this. So I talked about the laminae. It turns out that if you look at the cortex, you also have another type of organization that is called columnar organization. So in V1, this first visual region, you have what we call ocular dominance columns. What that means, so we also have orientation columns. And we can try to start to image these. And imaging these is really hard. We really have to push the scanner, and we can only scan a really very tiny part of the brain. And we really have to, you know, work together with the MR physics people that know their stuff to be able to push everything in this direction. The point is, what you get is if you imagine a very flat piece of V1, not only does it have depth, these layers, but it has a surface, right? And if you look onto the surface, it actually has columns. So this is a flat patch of cortex. There are different types of columns. These are the ocular dominance columns. So anything red here preferentially takes its input from the left eye, and everything blue preferentially takes its input from the right eye, right? So if you do this, you'll actually blank out the input to half of this, right? I forget which half because, yeah, but you see that. And it turns out that also for orientation, this is the case. So something oriented vertically, for example, would have a phase of 0. So that preferentially activates things that are red, patches that are red, whereas something horizontal would be something like greenish. Yeah? And so you see that there's like this beautiful sort of zebra stripy organization on the surface. And to measure this, we also need to have very high resolution imaging. But when you push everything, you can do this. And that's pretty cool, because this is a recent development. We've only been able to do this for a few years. And it really is starting to you know, bring a whole host of new questions into view. So I'm going to sit down a bit. Do I have more questions? No, because I only have 1 second left. I'm going to skip this. I'm just going to. Oh, really? Oh, I'm. Okay. Well, the torture continues. Thanks. Yeah. So you can. Oh, that's great. Yeah. So again. Nature isn't so easy on us, right? Nature really doesn't like us much. And because if nature liked us a lot, it would have created spherical heads and spherical brains that are beautifully flat and smooth and all that. It's not the case. If you look at a brain, it looks like this. It's all folded and crumpled up, and it's really hard to make sense of. And so one of the things that people have tried to do is then scan with, you know, you can change your voxels. They don't have to be cubes. They can also be like rectangles, like beams, like pencil voxels, we call them. And then you can try to scan them in one direction and then in the other direction. So you can really try to start pushing the scanner in really weird ways, but they allow you to sample the surface or the columns, so the layers or the columns of the cortex just right. So in our lab, what we're actually doing is we're scanning only lines. So we're not caring about 3D anymore. We only care about 1D. And so we don't scan the whole brain, we just scan a single line through the brain. And so my PhD student, Jurjen, will give a guest lecture near the end of the course. Well, he'll be explaining how to do this. But of course, then you need to know exactly where you're going to put this line, right? If you put it in the wrong place or in the wrong direction, you're not measuring anything that relates to laminae because you're just going in the wrong direction, right? So you have to really plan that line to be just right and just go through a patch of the cortex perfectly perpendicularly. And so we're setting up pipelines to plan this and to scan this, and he'll be showing you how we're doing that.
Does the architecture of the layers vary per area of the brain?
Yes, very much so. Yeah.
How do you know in vivo what the layers are when you're looking?
Yes, that's a good question. I think Jurjen will talk about this a bit, but what you see here is that the density of cells is different, right? What you see, so this also depends on the specific region. Still have 15 minutes, right? So here we go. In V1, you can find, so the V1 is a sensory region, right? So what's important for a sensory region is that it works with the incoming information very efficiently. Yeah? Now, efficiency in terms of neural processing generally is associated with myelinization, right? So that speeds up axonal transmission. Yeah, does that anybody know this? Everybody, like myelin goes around the axons, right? So there's a sheath around the axons. The more myelin, the faster the action potentials can travel along the axon. And so what this means is that in V1, its importance is in terms of processing incoming information. And what that means is that you can find a lot of myelin in layer 4 of V1. Yeah?
So are these images we see here, are those from an MRI?
No, this is histology. What? Sorry. Yeah. So, oh, here we go. Sorry.
Like from you take a pathology sample
Yes.
And you assume it's the same in vivo. Right?
Yeah, because so people are very similar. So the dead person is going to be similar to a living person. Yeah, the cells don't migrate tremendously. So there are beautiful atlases. There's a project called the Big Brain Project, where they've actually taken like one or a few human beings post-mortem and really sliced them up at the like 25 micron level and done this type of thing on it, and then created an atlas. So you can actually go online, find it, Big Brain Atlas, and then you will have like a super high resolution histological atlas of a brain. It's pretty gorgeous. Yeah. But with our specific anatomical sequences that we can scan with the MRI machine, we can actually see proxies of myelin. I don't know if you remember, but if you see a T1 weighted anatomical scan, the white matter is white and the gray matter is darker gray. And the white matter is white mainly because of fats. It's very high in fat, and that's because myelin is also a fat. So if you're looking at a T1 weighted image of high enough resolution and you're looking at V1, what you will see is a white line going through V1. You can actually pinpoint layer 4 in an anatomical scan. I can show you that in my brain. I'll do that next lecture. So if you have high enough resolution, like 0.6 or something millimeters, you can actually trace this line. And it's actually known from histology. It's called the stria of Gennari, from an Italian anatomist from like the 1800s or something. He already found this, and we can actually see it in the living brain nowadays. I don't know why I'm pointing here. So yeah, if you are ever a subject in one of our experiments, you can get that sort of thing. So conversely, if you're interested in, so this myelinization, you can also see in, for example, motor cortex. But what's motor cortex for? It's not for incoming information, for output. And that means that its myelin content is higher in another layer. And you can actually see this also in motor cortex. Not only is motor cortex myelinated in a different layer, it's also thicker. It's very easy to work with. But yeah, so you can actually do very detailed histology in scanners, in living human beings, because all of these different sequences, all of these different scans that we can perform in terms of anatomy, just, they intersect with the histology in some way, and then you can combine them and you can create beautiful quantifications of histology in that way. Also, quite a segue, but yeah. So you can imagine that if you have this very high resolution stuff, not only do you have a lot of data, but you also become very sensitive to small errors in terms of your analysis. There's so much detail here in terms of these layers and all of the convolutions. It's very, very complicated to work with. And that means that you can't use very standard analysis routines. You have to like develop everything yourself. You have to perform first the computational segmentation of the anatomy to find the boundary between the white matter and the gray matter. And then you also have to segment the peel surface, the outside of the white matter, right? Because all of those layers are in between those two surfaces. Now, this is something that doesn't really work well, automatically. Your brain is really good at doing it. Your visual system is really good at doing it. You can draw it. But to teach a computer to do it in sort of computer vision sense is really, really hard. And people are only now starting to leverage like convolutional neural networks to start doing this. But what you want is to have this sort of 3D mesh of both of these surfaces, and then you can start working with these. I'll go into how we do this and how we start using this type of representation also to visualize and work with our functional data. But yeah, there we are. But if you're looking at this if you're trying to really push the envelope in terms of fMRI, it becomes that there is really hardly any room for any error, right? So you really have to be very, very detailed in terms of any step of your analysis, both in terms of the analysis of the anatomies and in how you analyze the function. Yeah? This will start to make much more sense when we are there. Yeah. So, like I said, right, we have a scanner. It can do many different things. It's a very versatile machine. So I just want to get into how we can also scan different aspects of the blood response. So if you look at the layers of the cortex, you see that they are not just different in terms of their computational roles. They are actually quite different also in terms of their vascular structure. What actually happens is the blood comes in from the arteries from the outside, goes down, and then it drains up again. So there's a direction of blood drainage, like so. Yeah, it's always raining upward. That's really a Slayer song, isn't it? So you have blood being injected, oxygenated blood being injected down, and then it gets used somewhere in these layers, but there's always this sort of blood fall, waterfall going upwards. The vessels become larger near the top, closer to the peel surface, to the outside of the brain. And that causes a lot of problems for this specific laminar fMRI. In the standard way that we are measuring the bulge response, we have a bias for larger draining veins. You get beautiful signals here. You get not so beautiful signals here, because the veins here are larger, and the specific way in which we scan picks up the signals from these larger veins more. There are other ways that we can scan, and so in the physics lecture, we'll try to show you a bit about the difference between these two. But we have another way of scanning, which is called spin echo, which is less biased to these larger veins. So you don't have this bias where you're only recording stuff from there, but it also has a much lower SNR, right? So there is always this trade-off, right? How much do I value my specificity, and am I willing to torture my subjects for longer? Yeah.
What does SNR mean again?
Oh, sorry. Yeah, it's a signal to noise ratio. Yeah, it's the. Thanks for that question. It's a very important concept because you see this in any stage, right? So SNR, you can quantify the signal to noise ratio in anatomical images and functional images, and all that we are interested in many cases is to get as high as possible signal to noise ratio, right? Because the signals are what we care about, and the noise is what we want to suppress. So what does this affect? How does this affect our measurements? Well, if we start doing fMRI, laminar fMRI, that is, with these very, very small voxels, even if we don't expect any difference between the layers, what we get is tiny responses near the white matter and huge responses near the CSF. And we can't do anything about it. So if you expect something that's specific to the input layers of, in this case, V1, you expect something to activate more strongly here around layer 4. So you expect a hump here. But it's very hard to find that hump if there's already this enormous slope there, right? And so if we are pushing the boundaries of this laminar fMRI and really interested in the computational roles of these different layers, we are stuck with this in many cases. So this is from an experiment of ours that really showed me how shitty this can be. You live and learn. There are other sequences, again, different programs that the scanner can run where you are even less sensitive to this bias. You're actually looking at yet another property of this blood response. Another way of looking at it gives you less of a bias towards these PO surfaces. So here, this is the other way around. This is taken from motor cortex, and you see that the bold response is incredibly biased. There's a huge response towards the CSF. So that's this, you can see here. And that makes it really hard to see anything that's happening in these other layers. Whereas if you do something like this vaso, you actually get these discernible humps also in other layers, right? So the crucial part here, take away from this, is that this one, it's very hard to see anything here because this is so large. Here you actually see double humps, and so you get more specificity. But it does oftentimes mean that you need to trade off. So I know the person that scanned this. He put someone in the scanner for 4 sessions in a row, right? So you get lower SNR, meaning you need to acquire more and more data to average to increase the SNR, to lower the noise. And so if you're wanting to ask very, very specific questions, you first need to know what you're doing, right? So you have to tailor how you're scanning. And then you have to have a very good idea of how do you analyze this, and how can you optimize your experiments to find anything. Is that clear? Now I have a demo that I wanted to show you quickly. I'll try to show it on. So we have a server where we put all of our data. Oh, wait, there's also questions. Difficult to see what you're pointing at. Oh, yeah, sorry. I hope that the transitions helped for that. Was that just about it's about the double hump. So I'm going to point you to brain viewers like this a couple of times during the course. So this is just run in a website. It's a WebGL thing, and you can so we're looking at the surface of the brain. I think this is me again, and so this was computationally segmented, and now we can inflate. We can zoom in, and so what I'm going to do is zoom into the back of the brain where these visual regions are, and now I have run out of time, right? And so it's not showing anything. Maybe it's broken. So we can map out the visual system. So red here is a line that separates the first visual region from the second, and then same in green. We see that happening also in the other. So we can actually draw boundaries between regions of the brain. And I'm so hoping that it's not broken, because it seems like it's broken, and it's totally broken, right? It's not showing any data. Come on. Okay, I'll try to fix this before next. Oh, this is such a. So you can actually, you see that I'm going through depth. I'm going from the white matter to the gray matter. And if I were showing you actual data here, this is not, you're supposed to see data here.
projected on the brain as you're seeing it here. And we actually have mapped these ocular dominance columns. So you can actually, what you would see is blue patches and red patches. And this is really that left eye, right eye contrast that only exists in V1. But yeah, I should have tested this beforehand. I'm very sorry. Yeah. I'll fix this and I'll show you, I'll send you the link. Wow. That's bad. Ending on a note like this. Yeah. Yeah, I'll be crying tonight, that's for sure. But have a great weekend. Thanks. Yeah. See you on Wednesday. Oh no. Okay, I'll fix this and I'll get back to you on Wednesday for that. Yeah. From the double hump, you could see that, for example, a specific layer is receiving feedback from, let's say, another brain region. Yeah? So we want to see these double humps because we're interested in separating feedback responses, like expectation responses. And that's what we can tell. But we, of course, need the precision to be able to do that. And in our standard ways of doing that, we have very little precision.